Welcome to Chapter 27. I'm going to do a male summary of the reproductive system. So I'm not going to go heavily through all the structures. I'm going to focus more on the physiology that seems to give us problems. So here we have an oocyte, so this is an egg, and we have all of these sperm we can see that are trying to penetrate the outer coating of the egg, which is the zona pellucida. Basically, what I want you to see from this picture is you can have 100 to 1,000 sperm trying to penetrate the egg, but remember, we only want one sperm carrying 23 chromosomes to actually get to the egg or oocyte, which also has 23 chromosomes, so we can put those together, have a fertilized egg, and have a human embryo that has 46 chromosomes. Here is a sagittal view of the male reproductive system. For my class, I really want you to understand the flow of sperm. So make sure that you know where sperm is created. So if I said from the site of sperm production until it's ejaculated, you could follow that flow. So here we have the testes. Remember, inside the testes, we have seminiferous tubules. We'll have pictures coming up in just a second. So we're going to go from the testes, seminiferous tubules, epididymis, ductus deferens, which goes through the inguinal canal here, wraps around the posterior side of the bladder, and it widens. So this widening is called the ampulla of the ductus deferens. And then from the ampulla, we go into, if we're sperm, we go into the ejaculatory duct. And then we pass through the prostatic urethra, then the membranous urethra, sometimes called the intermediate urethra, and then through the spongy urethra, and we come out the external urethral orifice. So I wanted you to see this is kind of a long, complicated journey for these sperm just to exit the male reproductive system. Here's a nice sagittal cut of the testis. So 1, if it's 2, it's testes. Right, testis is 1. And we can see the seminiferous tubules. So all of these tubules are going to be creating sperm. Then that sperm is going to pass through these other tubes, and they give them names like retitestis, straight tubule, efferent ductule. But basically, from the seminiferous tubule here, all the sperm is going to be funneled into the epididymis. So seminiferous tubule funneled into epididymis. And they usually break up the epididymis into this is the head part. So the sperm, I'm going to do in red. So if we're creating sperm, they're going to get flushed into the epididymis from the seminiferous tubule. And then they have to travel all the way down to the tail of the epididymis. And then they're going to be able to exit via the ductus deferens. So one of the things that we'll see is in this section, in the head, we have immature sperm. And by the time we get down here to the tail section, as we move to the ductus deferens, this is where you're going to have mature sperm. Here, they're trying to show you a seminiferous tubule. And they're showing you cells off to the side. These cells are called interstitial cells. So these interstitial cells outside of the seminiferous tubule are the ones that are going to produce androgens, and in males that androgen is going to be converted to testosterone. Okay, we're going to talk about male sexual response. The first thing that's going to happen is erection, and this will be followed by the next slide, which is ejaculation. So what is erection? It is enlargement and stiffening of the penis from the engorgement of erectile tissues. So hopefully you know the erectile tissues are corpora cavernosa and corpus spongiosum. So there's 2 bodies of corpora cavernosa and only 1 body of corpus spongiosum. So normally, arterioles are constricted. So if the penis is flaccid, the arterioles are constricted, no blood flow into the penis. Okay, so next, upon stimulation, so some sort of sexual stimulation, it could be mental, physical, sight, smell, touch, or just thoughts. can activate the central nervous system and the parasympathetic neurons specifically. So you need to know that parasympathetic neurons are what cause erection. So, or the stimulation of parasympathetic neurons. Those parasympathetic neurons are going to cause nitric oxide release, which causes the relaxation of the muscle, smooth muscle, and the arterioles dilate. So they were constricted here. Now they are dilated, and when the arterioles dilate, the penis fills with blood. What specifically fills with blood? So blood flows into the corpora cavernosa, and when the blood flows in, it blocks venous drainage. So the blood-filled corpora cavernosa block venous drainage, so the penis stays filled with blood, and that is erection. Remember, we have the corpus spongiosum. This is another erectile tissue. It doesn't fill as much as corpora cavernosa, but its job is to keep the urethra open so that the sperm can flow out of the urethra. Okay, the second part is ejaculation. This is the propulsion of semen from the male duct system. So remember, semen is the fluid plus the sperm. And this is controlled by sympathetic spinal reflexes. So the ducts and accessory glands are going to contract and empty their contents. So when I say ducts, you should be thinking ductus deferens. Remember, the sperm have to come from the epididymis, right? So ductus deferens, we could be talking about prostate. We could be talking about bulbourethral glands. We could be talking about seminal vesicles. So all of those have to empty their contents. Bladder sphincter muscle is going to constrict, preventing the expulsion of urine. Remember, we do not want urine and sperm traveling in the urethra at the same time. Then we have muscles that are going to undergo rapid series of contractions, and the ejaculatory event is called climax or orgasm. I think it's funny, your book says the force of this is about 11 miles per hour. Okay, spermatogenesis. This is the sequence of events that produce sperm. So this is the entire process. We're going to see some subparts of this process. There's a lot of sperm words in this chapter, so make sure that you kind of know the real definitions and exactly what each word is talking about. So this is the entire process that it takes to produce sperm. Most cells in the body are diploid, meaning we have 2 of every chromosome, right? There's 23 pairs, so we have a total of 46 chromosomes. One of them you got from your mom, and one of them you got from your dad. So if this is my cell, and I'm going to just draw 2 chromosomes, this one came from mom, and this one came from my dad. We know this because we form gametes that are haploid. So if I look at an egg, it has 23 chromosomes, and if I look at a sperm, it has 23 chromosomes. So that when these fuse together or fertilize, we will end up with one cell that has 46 chromosomes that can grow from a fertilized egg to an embryo to a fetus, eventually to a newborn. So meiosis is the process in which you make gametes. So we have nuclear divisions in the gonads. So what we're saying is ovary and testis, I'm just listing one, but it can happen in both ovaries or both testes. This is the only place we have meiosis happening. So no other cells in your body do you want to have half the chromosomes, only in the ovaries and only in the testes, because they're going to make gametes. Those are the only organs that make gametes. So 2 consecutive cell divisions. We have meiosis I and we have meiosis II. It produces 4 daughter cells. The chromosome number is halved, and it introduces genetic variation. Okay, we're going to look at a cell, and we're going to see what it does if it goes through mitosis versus meiosis. So here in mitosis, we're going to start off with a chromosome, or a cell, excuse me, that has 4 chromosomes. So we have 4 chromosomes. We're going to duplicate them. We're going to copy them. So now we have 8. So you should be able to count up these, and you should see we have 8. We really have 4 with a copy of each. right? So this chromosome is copied, this chromosome is copied, this one's copied, this one's copied. So you really only have 4 chromosomes, they just doubled or replicated. So you end up having 8. Now that 8 is going to split into 4 and 4. So here we have 1, 2, 3, 4, 1, 2, 3, 4. We started with 4. We ended up with 4. And these are identical. So the daughter cells are identical to the mother cell, or the cell we started with. Again, don't get confused about male versus female. They'll usually, the mother of all things, right? So they'll say the mother cell is the cell that's starting the division. It doesn't have to be a mom cell, right? It can be any cell. And then they call the result daughter cells. Okay, so over here on the meiosis side, what is meiosis going to do? We still have 4. It's still going to replicate, copy, and make 8. So this cell should have 8. What's going to happen to those 8? They're going to split into 4 and 4, and then they're going to split into 2, 2, 2, and 2. So what's different about meiosis is meiosis has a first division, and then it has a second division. It divides twice. What else is unique? You start with 1 cell with 4, and you end up with 4 cells that have 2 each. So you go from 1 cell to 4 cells, and you have half the chromosome number. Another thing to look at that I'll bring up later is the way that the chromosomes line up in the middle. So do you see how in mitosis they line straight up and down, and then they split down the middle? The left side goes to the left cell, and the right side goes to the right cell. In meiosis, they kind of line up like shoes, like pairs of shoes. And then the left shoe goes to this side, and the right shoe goes to this side. So here's just a more detailed explanation of meiosis. You guys don't have to go through all of the phases. You should have an overall understanding of what's happening. So don't try to memorize all of this. If I said what's happening in meiosis, tell me the part that we need to know for chapter 29. We'll talk about prophase and we'll talk about crossovers. So in meiosis I, you start the division process. Remember, if it says 1, this is the first division of meiosis. Remember, there's 2. And what happens is we have this little crossover event where pieces of chromosomes are exchanged. So you can see here how the green guy has a little bit of the purple foot and the purple guy has a little bit of the green foot. Basically, you're swapping chromosome sections. So this crossover that's going to occur allows you to have genetic variation. So let's say the green is mom and she has blue eyes, and this is dad and he has brown eyes. Mom is short and dad is tall. So mom is short and has blue eyes, and dad is tall and has brown eyes. Is it possible for them to have a child that has blue eyes and is tall? Yes. So by swapping these chromosome pieces, you can have more genetic variation in your offspring. Now we have meiosis II. So we already completed our first division. So where should we be? If we had 46 cells, we copied them all, we had 92, now we split to 46 and 46. So this is where we are, 46 and 46. Now what do we have to do? We have to split these into 4 cells that have 23 chromosomes each. So we're here at our 46. What's going to happen? It's going to go through its second division, and the chromosomes are going to get pulled apart, and now we will have 4 cells that have 23 chromosomes each. Here at this prophase area, do you see how most of these are green and most of these are purple? It had to do with the cells lining up like shoes, right? I'm going to come back to this again. Okay, so here's our entire series or process that we talked about, spermatogenesis. So now we get to finally talk about the seminiferous tubules of the testes, how you're going to have spermiogenic cells that give rise to sperm. We're going to see a mitosis event happen first. Then we're going to see meiosis happen second. And then third, we're going to see something called spermiogenesis. This is when you have spermatids, so you could think of kind of immature sperm cells, and they're going to be become or develop into sperm. Here's what's happening in our seminiferous tubule. They don't really draw the entire structure. So this is that seminiferous, I'm going to abbreviate, tubules. And where are these? These are found inside the testis, right? So on the cortex, this edge part, you're going to see spermatogonia. These are stem cells. Remember, we said first thing that's going to happen is mitosis. Second thing that's going to happen is meiosis. And then the third thing that's going to happen is called spermiogenesis, where we take these early sperm and we turn them into more functioning sperm, or what look like more functioning sperm we call spermatozoa. So spermiogenesis is this entire process. So spermiogenesis includes a round of mitosis, complete meiosis, so meiosis I and meiosis II division, and then spermiogenesis. Let's walk through this. Here's the spermatogonia, change colors, spermatogonia, and it undergoes mitosis. So what did we say? Mitosis is when you copy yourself and you're identical, right? So identical copy, same number of chromosomes. So these two cells are identical copies to the spermatogonia, or spermatogonium for one. This type A cell is going to remain behind. It could be copied again later. This type B daughter cell is going to enter into meiosis. So now we have our 2N cell, and if it goes into meiosis, we should end up with this. So here's my 2N cell, and it copies itself and splits. Now here's the tricky part. It says N and N. If this is confusing for you and you want to use that notation, it's fine. The reason they call it N, you remember how we separated the shoes? We had 4 of every chromosome. Remember here they had to copy themselves. So we had 4 of every chromosome. And we said these shoes go this way, these shoes go this way, right? So what happens is you end up, and I kind of alluded to this, these were the green ones. And these were the purple ones. And we said all the green go to one side and all the purple go to the other side. This is why they call it N, because they're saying you really only have one type. You don't have 2 of every chromosome. You have one type that you just happen to have a copy of. If that is confusing to you, don't worry about it. I'm just trying to explain it so that we can understand why what I'm saying differs from what the textbook is showing you. That really, it has to do more with the type of chromosome, not how many there are. So what should you focus on? Mitosis happens first. Then we're going to start meiosis. We have one cell, it divides into 2, and then that cell splits again for meiosis II, and now you have 4 daughter cells that have half of the chromosomes of your original starting cell. Okay, let's look at the seminiferous tubule. Out here in this cortex area, this is where you would have the gonia, I'm going to abbreviate, spermatogonia, that is doing mitosis. And you're going to have a type A and a type B. That's happening out here. Then when that type B cell gets selected, splits again, and then splits yet again to complete the second division of meiosis, that's happening as you move away from the cortex. So these tails right here, these are all sperm tails. So the sperm here with all of their tails are closest to the medulla. So this is more towards the medulla or the middle of the seminiferous tubule. Basically, this slide is saying in words what we just saw in the figure. So we have the spermatogonia that's in the seminiferous tubules. I think it's I actually. So in the seminiferous tubules, the spermatogonia are going to divide, and this begins at puberty, and you're going to divide into a type A and a type B cell. The type A cell stays behind. It is a germ cell. Type B is going to move towards the lumen or the middle and develop into primary spermatocytes. We've seen this figure here. Here's the seminiferous tubule. Here's the lumen with the sperm tails. And we said out here, the cortex is where the spermatogonia turns into or divides by mitosis into the type A and the type B daughter cell. The type A sticks around. The type B daughter cell is going to enter meiosis. So now we have meiosis 1 and 2. That daughter cell, that type B daughter cell, is going to turn into or complete the first division, and you're going to have secondary spermatocytes. Then those cells are going to copy, and now you're going to have early spermatids. Eventually, they're going to shed their cytoplasm, and they're going to become what we would think of as sperm that have a flagella. And now we have our spermatozoa. And remember, this process was the spermiogenesis. So not spermatogenesis, but spermiogenesis. More words. Meiosis I, the primary spermatocyte. This is really the type B daughter cell from the mitosis division. But now we're in meiosis. That type B daughter cell is going to split into 2 secondary spermatocytes. Remember, if you want to call this 2N for right now, that's fine. Meiosis II, each secondary spermatocyte turns into a spermatid. And now we're back to the image again. Here's our primary spermatocyte, divides into secondary spermatocytes, splits again into early spermatids. So if you see the word spermatid, you should think half the number of chromosomes. At this point, it has finished meiosis II, and so it only has half the number of chromosomes. The third stage, so after we do mitosis and our 2 rounds of meiosis, now we're going to take those early spermatids and we're going to transform them into actual sperm or spermatozoa. So what's going to happen? The spermatids are going to have to elongate, they're going to have to lose their excess cytoplasm, and they're going to form a tail. When you look at sperm, they have specific regions. So they have a head region which has the genetic material. So you should be thinking DNA. They're also going to have an enzyme that can eat through or digest the zona pellucida, which is covering the egg. Then they have a mid piece, which has tons of mitochondria, and then they have a tail, which allows them to move, and that's called a flagellum. So this should make sense to you. If you need movement, why do you need mitochondria? Because mitochondria take glucose and turn it into ATP. What do you use the ATP for? For movement. Remember also that semen has fructose in it, and that fructose can be used, transformed, and modified into glucose or used in cellular respiration to produce ATP. It takes about 24 days to go from an early spermatid to what we would recognize as a spermatozoa or sperm. So you can see it has to lose that cytoplasm. We're going to have our mitochondria line up in that mid piece. You can see the DNA here in purple, and you can see this kind of cone, this acrosome, that's going to have enzymes in it that are going to digest the egg. Sustenacular cell, what is it? We saw our ring, our lumen. Here's our lumen. This is our seminiferous tubule. And we saw this yellow stuff in a previous slide. That is the sustenacular cell. I have another picture coming up. Remember, we have the tails, the sperm tails that were in the lumen. Also, I'm not sure if you remember seminiferous tubule, where we make the sperm, they're immature. Do you remember that first slide we said in the epididymis is where the sperm mature? So their tails don't work yet, So we have to have testicular fluid secreted by the sustenacular cell into the lumen to flush those guys into the epididymis. Also, they can, inside the sustenacular cell, you can have macrophages that can break down, phagocytize germ cells, anything that's wrong or faulty. So remember that germ cell is that type A daughter cell. If anything happens to it where its DNA is compromised, you can get rid of it within the sustenacular cell. Remember during spermiogenesis, when we have that cytoplasm, we're going from this nice round cell into kind of this fine-tuned cell, which is sperm, right? When we go from spermatids to sperm. So that cytoplasm that sloughs off. The sustenacular cell also provides nutrients, signals for the cell to divide. So we call it a supporting cell. It's called the sustenacular cell. Same figure, you can see that it's called sustenacular cell. It's everything in gold here. And this would just be a wedge of the entire seminiferous tubule. So you would have lots of sustenacular cells, right? Not just one. And so the sustenacular cell is going to provide kind of this area or space. It's going to, if this type A daughter cell becomes faulty, we can get rid of it. The excess cytoplasm also secretes fluid into the lumen of the seminiferous tubule, so we can push the sperm to the, oh, I forgot my I, epididymis. Another important function of the sustenacular cell I've highlighted here in red for you. Because sperm are not formed until puberty, they are absent during the immune system development. They would not be recognized as self. Remember how we talked about antigen presenting cells, and they take antigen to the thymus, and your T cells become educated, right? So that your T cells can learn what's a self cell and what's a foreign cell. Well, because sperm don't develop till puberty, if they weren't housed inside this sustenacular cell, if they weren't kept away from the immune system, your immune system would see the sperm as foreign and would try to kill them all. Under the right conditions, spermatogenesis, so that's the whole process, entire process, is going to take 64 to 72 days. Sperm are unable to swim, but the pressure of the testicular fluid pushes the sperm into the epididymis, where they gain motility and fertilizing power. So remember, they're immature at first. They have to go to the epididymis. And if you remember, when they come out of the seminiferous tubule, we said they go into the head, and then they come down, and they go into the tail, and then eventually up the ductus deferens. So that epididymis area is a maturation area for the sperm, and you can also think of it as storage for the sperm, because it takes quite a while from when we initiate that spermatogonia until we actually have fully functioning sperm. On to our favorite topic, hormone regulation. You should be able to look at this. It should make some sense to you. So they're saying hypothalamic, pituitary, gonadal axis. Do we remember that GnRH from the hypothalamus is going to trigger the anterior pituitary to release FSH and LH? Those are going to go to the gonads, right? And FSH is going to stimulate gamete production, and LH is going to stimulate hormone production. And if we're talking about males, that hormone would be testosterone. We haven't mentioned inhibin yet, and I'll talk about it in the following slides. Walking through the sequence of events: 1. Hypothalamus releases GnRH. 2. GnRH stimulates anterior pituitary to release FSH and LH. They have separate jobs, right? So 3rd, FSH is going to go to the sustenacular cell. Remember, this is in the seminiferous tubule of the testis. And it's going to cause the sustenacular cell to release something called androgen binding protein, or ABP. This makes spermatogenic cells, that's the gonia, right? Spermatogonia, receptive to testosterone. LH, 4th step, is going to stimulate the interstitial cells to release testosterone. So these two have to work together. The FSH is going to cause the sustenacular cell to release an androgen binding protein. It makes the spermatogonia receptive to testosterone, and then LH is going to stimulate the interstitial cells to release testosterone. And so what do you think happens when these two things come together? We're going to drive sperm creation. Step 5, testosterone is the final trigger for spermatogenesis, right? So for creating sperm. Rising testosterone levels cause a feedback, and we inhibit GnRH. If we inhibit GnRH, we also inhibit the anterior pituitary hormones. Secondarily, something called inhibin is released when your sperm count is high. This also inhibits GnRH and subsequently FSH and LH. Now we get to walk through all those words we just saw. So here's my hypothalamus. It's going to release GnRH. It's going to go to the anterior pituitary and cause the release of FSH and LH. The LH is going to go to the interstitial cells and cause the release of testosterone. FSH is going to go to the sustenacular cell, and it's going to cause the sustenacular cell to release androgen binding protein. So remember, this FSH is basically telling the spermatogonia, or making the spermatogonia receptive for testosterone. Now the testosterone comes in, and that drives the spermatogonia to undergo meiosis and produce sperm. So now what are they talking about, this negative feedback? They're saying if your testosterone level is high, or if your sperm level is high, that can inhibit GnRH, FSH, and LH. Turn it off. So if LH triggered your interstitial cells and you're making tons of testosterone, basically this elevated testosterone can go back and shut off hormone production. We've started the process that you told us to start. We're doing it. We're making testosterone. Don't tell us to keep making testosterone, right? It's kind of a conservation of energy. We're already doing what you told us. You can shut those off. What about sperm production? Well, the whole point of releasing these hormones was to drive sperm production. Now you've got tons of sperm being produced. The sustenacular cells are going to release something called inhibin, and inhibin can also go back and shut off GnRH and FSH and LH. So if your testosterone level is high, or if your sperm count is high, both of those are saying, we've done what you've told us to do. You know, message received. There's no point to keep releasing GnRH and therefore FSH and LH. Okay, well, this is the end of my summary for males. You can go watch the female video. If you have any questions, please email. Bye.